Usually when we talk about a medical diagnosis, there's this expectation of just absolute precision. It feels kind of like engineering.
Right. It's very binary.
Yeah. You break your arm, the x-ray shows that jagged white line of dense bone right against the soft tissue, and the doctor just points at the screen and says, there it is.
Exactly. Broken or not broken. I mean, the contrast is literally built into the physics of the machine.
It's clean, but if you zoom in on a tumor or you try to map out the complex tissues of a developing organ, suddenly that x-ray machine is totally useless. Yeah. You are looking at a cellular landscape for you that is, well, honestly, it's murky. Everything just blends together.
Oh, it is the absolute definition of diagnostic muddy waters. Without, molecular tags. You can't tell a malicious cancer cell apart from an immune cell that's just doing its job.
Yeah.
And that murky landscape is exactly what this new computational tool called Starfysh is trying to decode. We're looking at a fascinating paper from Nature Biotechnology today. And to understand how this tool is completely revolutionizing our view of complex diseases like cancer, you first have to look at how we currently try to map the microscopic neighborhoods of our bodies.
Okay, let's unpack this. Let's look at the current gold standard. We do this through a technology called spatial transcriptomics, or ST.
Yep, ST.
And the basic idea for you listening is mapping out exactly where specific genes are turned on or expressed inside a slice of tissue. We want to know what the cells are doing right this second, but there's a massive technical glitch.
A huge one.
The resolution is basically stuck in standard definition.
Exactly. I mean, ideally, we want to look at a single individual cell in total isolation, but ST can't quite do that yet on a massive scale. Instead, the tissue slice is laid over a glass slide, and that slide is divided into these tiny capture locations, which we call spots.
Right. Think of them as microscopic sticky pads that are just designed to catch genetic material.
Yeah, but physical limitations completely mess with the signal.
And the paper specifically mentions the lateral diffusion of RNA. Let's really dig into that mechanism for a second, because it's crucial. When researchers prepare a tissue slide, the RNA molecules, which are the chemical messengers telling us what genes are actively turned on, they can literally drift across the tissue slice while the chemical reactions are happening.
They just bleed into adjacent areas. It's like trying to take a photograph of a really dizzy street corner with a slow shutter speed.
Oh, that's a good way to put it.
Right. Each of these capture spots ends up collecting a blended mixture of RNA from multiple different cells. So when you look at the data for one spot, you aren't seeing one distinct tumor cell.
You're seeing a blur.
A total blur of tumor cells, immune cells, and structural cells, and they're all screaming their genetic information at the exact same time.
So if the spot's in a blended mess, how did scientists historically figure out what was actually inside them? I mean, if I have a blurry pixel, how am I figuring out what colors are actually making it up?
Well, before Starfysh came along, they had to bring in an outside dictionary to translate the blur. They relied on a completely separate data set called single-cell RNA sequencing, or cRNA-seq.
And the process for that was brutal.
Oh, absolutely brutal. They would take a piece of the tissue, use enzymes to literally dissolve the cellular bonds, just grind it up.
Literally grinding it into a soup.
Yeah, breaking it down into individual isolated cells, sequence them, and then use that pristine data as a cheat sheet.
So the logic was kind of like, based on this separate, perfectly clean list of cells we ground up yesterday, this blurry spatial spot on today's slide probably contains 20% T cells and 80% tumor cells.
Precisely. It's a mathematical process called deconvolution. But that approach has glaring fundamental flaws. First off, grinding up tissue destroys the very spatial context you're trying to study.
Right. You know the cells exist, but you have zero idea where they lived in relation to one another.
Exactly. And second, what if you only have a tiny clinical core biopsy from a patient?
You can't just grind half of it up to make a reference dictionary. I mean, a pathologist needs every millimeter of that tissue to make a life or death diagnosis.
You absolutely cannot. And relying on a completely separate data set introduces massive computational biases.
Oh, so?
Well, if your dictionaries was built from a different patient or even a different physical zone of the exact same tumor, the cell densities and biological states just won't match perfectly. It introduces what we call batch variation biases.
Okay.
The older tools, systems like DESPI, Tangram, or Bayes Prism, they all rely on these external single-cell references. They carry external biases into every single calculation.
Which is the core problem Starfysh solves, right? Because Starfysh operates entirely reference-free.
Entirely reference-free, yes.
Which brings us to the math. How does Starfysh actually figure out what cells are in a blended spot without an external dictionary? If I don't have the cheat sheet, how am I translating the language?
It relies on a mathematical concept called archetypal analysis. Think about standard data clustering. Usually, algorithms take a bunch of blurry data points and average them out to find the middle ground.
Right, finding the generic mean.
Starfysh does the exact opposite. Instead of looking for the average, it scans the tissue map for the most distinct, extreme gene expression profiles. It actively hunts for the purest spots in the entire sample. And these extreme points become mathematical anchors.
Okay, I'm going to use an analogy here. Imagine a tissue spot is a blended fruit smoothie. Previous methods forced you to go to the grocery store, buy a separate perfect strawberry, bring it back to the lab, and compare it to the smoothie to figure out the recipe.
Right, that's the strNA-seq dictionary.
Exactly. But Starfysh instead just looks around the inside of the blender itself. It searches for the microdroplets that taste the absolute most like strawberry or the most like banana. It uses those extreme points already inside the mixture to reverse engineer the entire recipe of the smoothie.
That is a brilliant analogy. And that reverse engineering happens through a deep generative model.
Okay, lay the deep algorithmic detail on us.
So the algorithm uses what we call known marker gene sets as a starting point. These are basic universal lists of genes we already know belong to broad categories like T cell or tumor cell. In the math, this is applied as an empirical prior. Specifically, they use a Dirichlet distribution prior.
Wait, let me push back on that for a second. If Starfysh is starting with a list of known markers, isn't it still relying on previous knowledge? Like, isn't that just another cheat sheet? How is it discovering anything new?
That is a great question. And it's exactly where the genius of combining the Dirichlet prior with archetypal analysis comes in. Think of the prior as like a gentle gravitational pull.
Okay.
It gets the mathematical model looking in the right general direction, say, pointing the telescope toward a specific galaxy of tumor cells.
All right.
But the archetypes, those extreme points found in the actual patient's tissue, they adapt the final coordinates.
So if a patient has a tumor cell that mutated in a completely bizarre, undocumented way, the generic tumor list gets the algorithm in the neighborhood, but then the algorithm snaps to the actual physical reality of that patient's unique mutation.
Exactly. It updates the anchor to match the patient's unique biological reality. It allows Starfysh to characterize highly context-specific cell states that have never been cataloged in any reference database. It learns the patient's specific biology on the fly.
That is wild. Okay, so we're about 5 minutes in. Let's do a quick recap and really bullet summary for you to consolidate all this dense information.
Those are good.
Bullet 1, spatial transcriptomics, or ST data, is inherently blurry because of RNA diffusion.
Right.
Bullet 2, old computational methods needed a separate, expensive cheat sheet to unblur the data, which introduced massive bias.
And destroy the spatial context.
Exactly. And bullet 3, Starfysh finds the purest spots, the archetypes already in the tissue, to mathematically unblur the map, completely reference-free.
You've got it perfectly. But, decoding the genetic ingredients of the smoothie is only half the problem. You also need to understand the physical structure of the container it's sitting in.
Which leads to Starfysh's second major breakthrough. It doesn't just look at the genetic data, it actually integrates the visuals, it looks at the pictures.
Right. What's fascinating here is that when pathologists look at a biopsy, they stain it, usually with hematoxylin and eosin, or H&E staining.
That's what turns the tissues all pink and purple on the slides, right?
Exactly. And this reveals the actual shapes, sizes, and physical structures of the cells. Surprisingly, most of the older computational tools completely ignored these photographs.
Wait, really?
Yeah. They basically threw the visual data in the trash when doing the math, treating the tissue like an abstract spreadsheet of genes.
But how does Starfysh actually merge a high-res photograph with genetic data? I mean, they're completely different languages. One is a picture of pink blobs. The other is a literal list of RNA molecules.
It utilizes a deep learning architecture called a product of experts, or POE, and it integrates that with a deep variational information bottleneck.
Okay, that's a mouthful. Let's break down that mechanism.
Let's do it. So the model uses a visual neural network, specifically a ResNet 18 encoder, to mathematically flatten the image patches into data points. It takes the curves, the cell density, the gaps in the tissue, and turns them into numbers.
Let's clarify that bottleneck concept for you, because product of experts sounds incredibly abstract. Imagine two detectives working on the exact same suspect.
Okay, I like where this is going.
Detective 1 only has access to financial records, that's the gene expression data. Detective 2 only has access to security footage, that's the H&E image batches. But they aren't allowed to just hand each other their files. They're placed in separate rooms and told they can only pass 3 numeric coordinates to the police chief to describe the suspect.
That is a perfect visualization of the information bottleneck. By restricting the flow of information into a shared, low-dimensional mathematical space, both detectives are forced to discard all the useless noise, the fluff, the imaging artifacts, the genetic static, and only pass the absolute most critical, condensed clues.
And when they hand their coordinates to the chief, those numbers have to agree.
That is the product of expert part. By mathematically multiplying their probabilities together, Starfysh parses the gene data and the visual data to agree on a single, unified reality.
So if the genes say there's a dense tumor here, but the picture shows empty connective tissue, the model forces a recalculation until both realities align perfectly.
Exactly.
But why is the picture so critical? I mean, if we know exactly what genes are turned on, if we literally have the molecular blueprint, what does a photograph actually add to the equation?
The image provides structural context that genes alone cannot explicitly confirm. It tells Starfysh about cell density. It shows whether cells are packed tightly in a rigid tumor core or spread out sparsely in healthy connective tissue.
It gives it the architectural reality.
Right, and it highlights spatial dependencies because cells physically touching each other heavily influence each other's behavior. By forcing the genetic predictions to obey the physical geometry of the image, the deconvolution accuracy just skyrockets.
And the researchers proved this by benchmarking it, right?
It did. They tested Starfysh against a newer, highly expensive technology called Xenium, which actually does map at true single-cell resolution.
So Xenium is the actual answer key.
Yes. And when they ran Starfysh on blurry standard ST data and compared its predictions to the ultra-clear Xenium data from the exact same tissue slice, Starfysh consistently outperformed all the older tools, DestViz, Tangram, Bayes Prism. It synthetically recreated the high-definition reality.
That is incredible. So we have this highly accurate, high-definition lens that merges genes and geometry. Now, what happens when we point this tool at one of the most complex terrains imaginable?
We look at breast cancer.
Exactly. The researchers analyzed tissues from several types, ER-positive, triple-negative breast cancer, and a very rare, highly aggressive subtype called metaplastic breast cancer, or MBC.
And here's where the biology gets incredibly complex, because cancer isn't just a monolithic lump of bad cells.
Right, it's not a monolith.
It is defined by intratumoral heterogeneity. The cells inside a single tumor are constantly mutating, adapting, and essentially breaking off into different warring factions.
Yeah, and when Starfysh analyzed just a single triple-negative tumor, it successfully mapped 29 diverse cell states. We're not just talking about tumor cells versus healthy cells here.
No, not at all. We're talking about multiple distinct evolutionary branches of tumor cells, various T cells, macrophages, endothelial cells, all interacting in completely different physical zones.
And mapping those zones led to a massive biological discovery in the paper. They documented something called a spatial gradient, or a pseudospace. Let's really dig into what that actually looks like in the tissue.
It completely shatters the idea that tumors exist in discrete, static states. Starfysh revealed a continuous physical transition flowing across the tissue. On one side of the tumor, the cells were what we call basal-like, a pretty standard classification of breast tumor cell.
But as you physically drag your focus across the tissue map?
Right. The cells transitioned along a gradient. They gradually lost their basal properties and acquired highly aggressive MBC-like properties.
They were actively mutating across physical space.
Yes.
Here's where it gets really interesting. As they mutated, they started undergoing EMT epithelial mesenchymal transition.
A crucial mechanism.
Yeah, for context, EMT is a biological process where a stationary structured cell completely changes its identity, tearing up its anchors to become a mobile invasive cell. It is the hallmark of cancer preparing to metastasize and spread through the body.
But the truly insidious part isn't just that the tumor cells change themselves. If we connect this to the bigger picture, is that they actively change their neighbors.
Oh.
Yeah. The Starfysh mapping showed that as these tumor cells advanced into that aggressive EMT state, they began surrounding themselves with very specific immune cells.
Specifically, regulatory T cells, or TREGs, and M2 macrophages. Why those two specific cells?
Because of their physiological functions. TREGs act as the brakes on your immune system. Their normal job is to stop your killer T cells from attacking your own healthy organs.
Okay, and the M2 macrophages?
Those are your body's tissue repair cells. They show up to heal wounds, clear debris, and actually build new blood vessels.
So the tumor is essentially brainwashing the body's medics and peacekeepers.
Exactly. The tumor is intentionally shaping an immunosuppressive niche. By surrounding itself with TREGs and M2 macrophages, the mutating tumor builds a biological moat.
A fortress.
A fortress. It creates a microenvironment where the body's natural killer cells are commanded to stand down and where the body's repair cells actually start building blood vessels to feed the tumor. It allows the aggressive EMT state tumor cells to completely thrive.
That is, frankly, terrifying, but amazing to actually see mapped out. Okay, we're at the 10-minute mark. Let's do another quick recap and bullet summary for you.
Let's do it.
Bullet 1. Tumors don't just exist in one state. They exist on a shifting physical gradient.
Yep, the pseudospace.
Bullet 2. As tumor cells mutate into more dangerous invasive forms via EMT, they actively re-engineer their local neighborhood.
Bullet 3. They build a biological moat of immune-suppressing cells like TREGs and M2 macrophages around themselves to guard against the body's natural defenses.
Perfect. Which naturally leads us to the problem of intertumoral heterogeneity. If Starfysh can see these neighborhoods in one single tumor, can it compare neighborhoods across different patients to find universal targets for drugs?
Historically, comparing two different patients' spatial data was an absolute nightmare. The batch effects, like, the minor differences in how the clinical slides were prepared, or just the inherent biological noise of different humans, it made it like trying to compare a map of New York to a map of London when all the street names are scrambled.
Just noise upon noise.
Exactly. But because Starfysh uses those mathematically derived archetypal anchors and that deep generative model, it standardizes the math across different samples. The researchers successfully integrated 14 different spatial samples from 10 different patients.
Which is over 37,000 individual spatial spots mapped into a single shared mathematical space.
37,000 microenvironments compared side by side.
So what did they find when they looked at that highly aggressive metaplastic breast cancer across all those different people?
They found that MBC has wildly higher patient-to-patient heterogeneity than other types of breast cancer, like TNBC or ER-positive. Every single MBC patient's tumor was structurally a completely unique beast.
But despite that uniqueness, Starfysh found the structural weak points, right? It found the shared mechanisms running beneath all that chaos.
It did. Across these wildly different MBC patients, the tumors universally shared an enrichment in the P53 and PI3K AKT signaling pathways.
And what do those pathways do?
They regulate cell growth and survival, and their enrichment signals massive metabolic reprogramming. The tumors were ramping up glycolysis.
Which is burning sugar for fast, inefficient energy.
Right. And they were activating HIF-1 signaling, which is a specific pathway that allows cells to survive in extremely low oxygen environments, like deep inside a dense solid tumor core.
Wow. Why does this matter to you listening? Because identifying these specific spatial hubs means we can target them. And the most crucial detail the model found is how the tumor actually communicates with that defensive moat we talked about earlier.
Yes. The paper highlighted a specific communication channel between the tumor cells and those protective TREG cells called the FGFR signaling pathway.
Let's explain how this acts as a biological phone line.
Think of a signaling pathway like a chemical radio broadcast. The tumor cell releases specific proteins into the space around it. The TREG cell has receptors and antennas specifically designed to catch those proteins. When the FGFR proteins bind to the TREG receptors, it transmits a signal that physically alters the TREG's internal behavior, basically ordering it to suppress the immune system and protect the tumor.
But if we know the tumor is using the FGFR radio frequency to coordinate its defenses, we can jam the signal. We can literally cut the phone line.
And that is the ultimate clinical payoff here, moving away from carpet bombing the body with chemotherapy and moving toward highly targeted, rational drug design based on the physical architecture of the tumor.
So the papers suggest that FGFR inhibitors, or drugs that specifically target that altered glucose metabolism, could be future therapies for breaking down these hubs in MBC.
Exactly.
And it is vital to note that Starfysh isn't just for breast cancer. The exact same computational logic applies to mapping any complex tissue.
Oh, absolutely. The paper showed it mapping the intricate layers of the mouse brain, easily identifying the distinct cortex layers and the hippocampus.
They also mapped the delicate structures of human lymph nodes, right?
Yes. Finding the exact geometrical zones where immune cells literally train to fight infections. And perhaps most impressively, they tracked prostate cancer in patients undergoing androgen deprivation therapy.
AD therapy.
Yeah, this is standard treatment, but tumors often figure out how to resist it. Starfysh tracked the microenvironment changes over time and identified a specific spatial hub that actively resists the therapy.
And what was that resistant hub made of?
It was incredibly rich in cancer-associated fibroblasts, or CCAFs.
CCAFs.
Yeah.
And fibroblasts are basically the body's structural builders, right?
Yeah.
They lay down the collagen framework for tissues.
Exactly. So the tumor's resistance wasn't just the cancer cells magically mutating to become immune to the drug. The tumor was actively retreating into a protective bunker made of thick, fibrous CCAF cells. The architecture itself was the resistance.
The neighborhood protects the tumor. That keeps coming up as the underlying reality of this entire deep dive.
It really does.
So what does this all mean? Let's provide a final distillation. Starfysh is effectively a new computational lens. It takes the blurry, overlapping data of spatial transcriptomics, and without needing a single cell reference sheet, it brings our cellular neighborhoods into razor-sharp focus.
Using archetypes and histology.
Right, by combining archetypal analysis to find the purest extremes and deep generative models that force genetic data to agree with the physical reality of histology images, it lets us read the tissue like a highly detailed topographical map.
It reveals not just what cells are present, but how they communicate, how they build protective moats, and how they organize themselves in both health and disease.
Which leaves us with a fascinating question to consider for you to mull over. If tissues are truly ecosystems, and diseases like cancer rely on building these very specific neighborhoods,
Like surrounding themselves with tregs or hiding in a bunker of fibroblasts.
exactly. What if we could use tools like Starfysh proactively? What if we could map the spatial transcriptomics of our bodies long before we even feel sick?
That is the dream. Could we predict a biological neighborhood's collapse or spot a tumor laying the literal foundation for its fortress simply by watching the microscopic architectural shifts in our cellular cities?
It changes everything about how we view disease, not just as a broken part, but as a shifting landscape.